Bismillahirrahmanirrahim. We start the topical on the enzymes MCQ. And first I will run through the paper so that you can do it without the mark scheme, and then you can do it with the mark scheme. Now let's look at question number 1. The enzyme invertase catalyzes the breakdown of sucrose to glucose and fructose. We know sucrose is a disaccharide broken down to glucose and fructose. Three different enzyme inhibitors, X, Y, and Z, were investigated. The percentage inhibition of invertase was measured at different concentrations of an inhibitor. So when you had 2% inhibitor, now I've labeled the y-axis. This was not labeled in the question, but I've done it myself. So you can see at 2% concentration, the inhibition in Y was 3, and in Z was 5, and in X was 15. So always put some figures on the y-axis, and if not on the x-axis, then also put some figures on the x-axis. So what you have to understand is that, I've given the figures here, x at 2, then I drew a line at 2, then I drew a line at 4, and then I figured this out is how much was the difference between it. Now you can tell that the inhibition has increased in all three of them. So now you have to realize if the inhibition increase, so invertase is inhibited, so less glucose and fructose will be made. Invertase is inhibited, so less glucose and fructose will be made. Now, what are the valid conclusions from these results? From these results, the higher the concentration of inhibitor X, the less sucrose is broken down. That's correct. More inhibition, less sucrose broken down. So 1 was correct. Now let's look at 2. Why is 2 wrong? The answer is of course 1 only. The production of glucose and fructose using inhibitor Y is higher than when inhibitor Z is used. The production of glucose and fructose in Y is higher. Production of glucose and fructose means what? Less inhibition. But Y, you can see Y is inhibiting more. The inhibition is 25% if you just see it here, this side. And Z is inhibiting it only 5 or 6%, 7%. And Z, you see they are comparing Y and Z. Y is inhibiting it 25%, Z is inhibiting it only 6%. And the production of glucose and fructose at an inhibitor concentration of 2 arbitrary units is lower. Inhibition is lower. How is inhibition lower than at an inhibitor concentration of far than for all inhibitors? You have to realize is that the inhibition is increasing, so production of glucose is going to decrease. They've given it the other way around. As the inhibition increases, the production of glucose and fructose will decrease. So it is saying inhibitor concentration of 2 arbitrary units is lower. So production of glucose and fructose is lower. No, production of glucose and fructose is more at a lower inhibition. At 5% inhibition, more glucose and fructose is being produced. At 25% inhibition, less fructose and glucose is being produced. So please understand this is not very easy. Then coming on to question number 2, the following statements are about enzymes. Folding of an enzyme molecule causes the formation of the active site. The shape of the active site changes to enable the substrate to bind. Temporary bonds hold the substrate in the active site. More enzyme substrate complexes are formed at the optimum temperature. So, the following which statements are correct for the induced fit hypothesis, not the lock and key. So, which statements are correct for the induced fit hypothesis? That was 2, 3, and 4. The shape of the active site changes. Temporary bonds hold the substrate in the active site. More enzyme substrate complexes are formed at the optimum temperature. So that is why the answer was C. Then part 3 was a direct question. How is the Michaelis-Menten constant used? To assess the efficiency of an enzyme in catalyzing a reaction, to compare the affinity of enzymes. Yes, that's important. You compare the affinity of enzymes to their substrate. To find the maximum velocity of an enzyme, Vmax, no, that was wrong. To find the rate at which substrate is loaded by an enzyme, no, that was wrong as well. And these three were wrong. Then number 4, the graph shows the results of an investigation into the effect of amylase on starch at 3 different temperatures. Now you see graphs are very critical here. So concentration of reducing sugar. So as I said, just put some figures here. 5, 10, 15, 20. So concentration of reducing sugar was 5, then it was 10, then 15, then 20. Now read off the graph. At 70, the concentration of reducing sugar is become constant now at 70 degrees Celsius at a very early, at about something like 12. At 60, the concentration of reducing sugar has become constant at 15. At 50, the concentration of reducing sugar has become constant at 19. Concentration of reducing sugar. 10, 11 arbitrary units, 15 arbitrary units, 20 arbitrary units. Now it says which statements are correct conclusions using these results. The optimum temperature is 50. No, how can we say the optimum is 50? We haven't tried 40, 30, 20, 10. We don't know. We've just used three temperatures. The initial rate of reaction is highest at 70 degrees. Yeah, that's correct, because the initial rate of reaction is higher in the beginning. The higher the temperature, the more quickly the enzyme denatures. Yes, because the concentration of reducing sugar becomes constant. So that's why the answer was 2 and 3, and this was the answer. Then coming to question number 5. Substances called furanocoumarins are found in some fruits where they act as inhibitors of the enzyme CYP3A4. The enzyme is needed to break down toxins in the human digestive system. So dangerous level of these toxins may develop if these fruits are eaten. What does this information suggest about molecules of the enzyme CYP3A4? Now, first, we need to understand this question and then we go proceed. The furanocoumarins are found in fruits, and the furanocoumarins act as inhibitors of the enzyme CYP3A4. Now, this enzyme CYP3A4 is needed to break down toxins in the human digestive system. So basically, God has given this to us as an enzyme to break down the toxins in the human digestive system. So danger levels of these toxins may develop if these fruits are eaten. Now, if you add these fruits, the fruits contains the inhibitor, so the toxins are not going to be broken down, and the toxins will increase in the system. So now it says, what does this information suggest about molecules of the enzyme CYP3A4? So they lower the activation energy of the toxin breakdown process. Yeah, naturally. They break down the toxins. So what do enzymes do? They lower the activation energy. So this is how you have to understand is how the answer was A. The other possibilities were they show specificity for a particular substrate found in fruits. No, they will change permanently when acted upon by furanocoumarin. No, they will resume normal activity when levels of furanocoumarin decrease. No, they are an inhibitor. So this is what you have to understand about this question. Then a very direct question 6. In the two investigations, the rate of enzyme control reaction was measured at the presence of either a competitive or a non-competitive inhibitor. So competitive and a non-competitive inhibitor. What could be the effect of increasing the substrate concentration? Now, if you remember, the enzyme reaction starts off like this, and then it reaches a Vmax, and then it levels off. Now, when you add a competitive inhibitor, it decreases, but then as you increase the substrate concentration, it nearly levels off. So this would be substrate concentration on the x-axis. So it says, what could be the effect of increasing the substrate concentration? If you increase the substrate concentration, competitive inhibitors affect, so rate of reaction with competitive inhibitor will increase because the inhibition decreases. So with competitive inhibitors, what will happen is the rate of reaction will increase because when you add the competitive effect of increased substrate concentration, when you increase with competitive inhibitors, ultimately the competitive inhibitor, poor thing, is just sidelined. Non-competitive inhibitor, there's going to be no change. Now look at this situation. There are 100 active sites. There are 10 of these. 10 is the substrate and 10 is the inhibitor. So of course they're both competing for the active site and they can both fit in, but when this fits in, no product is formed. But when the blue fits in, product is formed. Now look at how I'm going to change the situation. If you're going to increase these 200, then there are 10 of the inhibitor, so this stands no chance now. But if you increase it even more, 1000, then to 10,000, poor of these, they will not get any lift. So they will be ignored because you have 10,000 of these. So now you look what I've done. When you increase the substrate concentration with a competitive inhibitor, the rate of reaction will increase. But with non-competitive inhibitor, it's not going to increase. In non-competitive inhibitor, you have a totally different situation. In a non-competitive inhibitor, you add something to the other site and the active site changes. So if you've added 10 active sites have gone. So you now only have 90 active sites. So by increasing the substrate concentration, the rate will not increase, it will become constant because now you have 90 active sites. 10 you have destroyed because 10 non-competitive inhibitors you have added. So 10 enzymes, their active site has changed. Out of 100 minus 10, only 90 are left. So 90 will keep on working. So even if you increase the substrate to 10,000, it's not going to matter. All the active sites will be occupied. Question 7. Two enzymes X and Y were used in an experiment. Enzyme X was from bacteria that live in rivers and lakes at temperature from 5 to 20. Enzyme Y was from bacteria that live in hot water springs at temperature 40 to 85. The experiment measured the concentration of product produced, starch, amylase, maltose, product maltose, by each enzyme at temperatures between 0 and 100 for after 5 minutes. Which graph shows the results? Now you have to be very careful when you look at this sort of a question. Now as you can see, I put figures on the graph. So I have got on the x-axis I put some figures and on the y-axis I put some figures. So I put in concentration of product 10 and 20. Similarly on all the y-axis I put 10 and 20, and then I put the 50 on the x-axis as well because we were given 0 and 100. Now you realize why each enzyme temperature. Now the experiment measured the concentration of product produced by each enzyme at temperatures between 0 and 100 after 5 minutes. So we kept them at 0, then we waited 5 minutes and we measured the concentration of the product, and then we did it at 5 and then 10, and then so the temperature was slowly increased and we kept on measuring the concentration of the product. So as it reaches the optimum, the concentration of product should increase. So now you could see that both the x in here and the x here were the same, but the y had to be, it couldn't be exactly between 60 and 100. It was less than 100. This was, you can say it's even less than 100. The optimum temperatures between was, they were temperatures of 40 to 85. So 40 to 85 means that we need to have some activity at 40. And here we have no activity at 40 for y. So read the question carefully. Put these, I'm trying to train you for any question which may possibly come in the exam. So it's not only this question. Question 8. Which statements describe some enzyme actions? Enzymes hold reacting molecules in such a way that their reactive groups are brought close together. In an enzyme catalyzed reaction, more molecules have sufficient energy to react. In the absence of the enzyme, yes, they lower the activation energy. Reactions catalyzed by enzyme take place at a lower temperature than they would without the enzyme. So yes, 1, 2, and 3 all were correct. Then question number 9. The diagram shows the mass of product. Starch, amylase, maltose. Mass of maltose formed over time in 3 reactions using the same substrate and enzyme. The volume of substrates, enzyme, and temperature were kept constant in each reaction. And you have these 3 graphs. Which statement explains the difference in these reactions? Now again, put some figures here. 1, 2, 3, 4, 5, 6, 7, 8, something like this. So you can put 10, 20. Mass of product. Now put that here, 10, then 20, then 30, and so on. Or you could put it anything. You can put 5, 10, 15 as well. So it says, which statement explains the difference in these reactions? The pH in reactions 2 and 3 has denatured the enzyme. No, how do we? That doesn't explain from the graph. There is non-competitive inhibitor present in reaction 3. No, non-competitive inhibitor cannot level out. There's a time. This has really reached the same. So there is the highest concentration of enzyme in reaction 1. Because you see what has happened, mass of the product has very sharply increased and then the mass has remained constant. But if you look at any point in time, the mass of the product is less. So 3, the mass of the product is less, 2, the mass is even lesser, and 1, it is the maximum. So mass of the product is dependent on the number of active sites. So this is how you have to figure this out. Please understand this. I always ask you all to label the axes and then read the story to yourself. Then question 10. Tyrosinase is an enzyme that catalyzes the conversion of the amino acid tyrosine into the black pigment melanin. Now this, of course, you will also study this in A2. It is responsible for the black fur color of some rabbits. A group of rabbits kept at 30 degrees resulted in 90% of the rabbits with light fur color. A second group of rabbits were kept at 10 degrees resulted in 90% of the rabbits with black fur color. One had light fur color and one had black fur color. Which hypothesis is supported by these results? An inhibitor is present in rabbit skin cells that can bind strongly to tyrosinase when the external temperature is 30 degrees Celsius. Yeah, that was the correct answer. But why does the other three wrong? At 10 degrees external temperature, there are fewer tyrosinase-tyrosine complexes formed and less phenylene is produced. Tyrosinase is an enzyme that is coded for by a gene that is switched off when the external temperature is 10 degrees Celsius. Tyrosinase is a temperature-sensitive molecule that is only activated when the external temperature is 30 degrees Celsius. Well, of course, you can see how the other are all wrong and A is the correct answer. So basically, the question says tyrosinase is an enzyme that catalyzes the conversion into the black pigment and is responsible for the black fur. So if tyrosinase is working, the rabbits are going to have a black fur. If it's not working, they're going to have a light color. So at 30 degrees center, they weren't working, they were light colored. So that means there was an inhibitor present which disabled the tyrosinase. So very easy from the question. Tyrosinase works, rabbits are black. Tyrosinase does not work, rabbits are light colored. So 30 degrees, the rabbits were light colored. So what has happened? Inhibitor is present in rabbit skin cells and bind strongly to tyrosinase when the external temperature is 30. Question 11. Which levels of protein structure are always involved when competitive and non-competitive inhibitors bind to enzymes? So both are tertiary structures because that's the final folding which results in the shape, and that's where the inhibitors bind to it. Question 12. Which statements are true about the optimum temperatures of all enzymes? It is the temperature at which the enzyme works best. Yes. It is the highest temperature at which the enzyme will work. No, they even work after the optimum. Like for instance, human beings have an optimum, so that's how the graph comes out. So if this is the optimum, which is 37, they're still working till about 45, but the rate is decreasing. It is between 25. No, all enzymes are not between 35 and 40. There are enzymes which work best at even 70, 80, 90 thermophilic bacteria. Question 13. Proteases are a group of enzymes that digest proteins. Which statement of protease is correct? A bacterial protease that is secreted from a disease-causing bacterium could act as an antigen and cause the production of an antibody. Which statement about proteases is correct? So yes, it's a protein. So if a disease-causing bacteria produces it, it acts as an antigen cause because it has a specific shape. The rest you can understand they're not the answers. I'm not going into the detail of that. Then coming to question number 14. Which words from the table correctly complete the paragraph about enzymes? Very simple. When the pH of an environment is decreased below an enzyme's optimum pH, dash bonds between adjacent dash groups holding the dash structures are disrupted. So when the pH of an environment is increased below an enzyme's optimum pH, comma, hydrogen and ionic bonds between adjacent R groups holding the tertiary structure are disrupted. So basically when you change pH, ionic mainly, but hydrogen bonds are also broken. So between adjacent R groups holding the tertiary structure are disrupted. Question 15. Lipase is a digestive enzyme produced by the pancreas that catalyzes the hydrolysis of dietary lipids. The table shows how the pH of a liquid food containing a high proportion of lipids decreases over time. Yeah, because you see lipids are fats or triglycerides. They're broken down into fatty acids and glycerol. So fatty acids are going to lower the pH. So pH of a liquid food and then time in minutes, and you can see how the pH is decreasing from 8 to 7 and then going down, really down to about 6.5. Which statements are possible explanation of the results of the experiments between 50 and 60 minutes? So this time, It's only talking about this time, in which now the pH is remaining constant at 6.5. Enzyme concentration becomes the limiting factor, substrate concentration becomes the limiting factor. Yes, no more fatty acids are being produced, so all the substrate is being finished. Then, as all the enzyme active sites are saturated, denaturation of the enzyme by the product, the pH is lowered, so denaturation of the enzyme is taking place by the fatty acids, which are resulting in lowering the pH. Products are acting as competitive inhibitors. The product could be sort of acting in, could then be fitting there and acting as a competitive inhibitor. That's why the rate, no more fatty acids are being made, so they are competing, and products are acting as competitive inhibitors. Fine. The AIDS virus produces a long polypeptide. The AIDS virus produces a long polypeptide that is hydrolyzed by a protease enzyme, producing several smaller peptides. So there's a polypeptide, and there's a protease enzyme which breaks this up into pieces. So this is going to happen. So it's hydrolyzed by a protease enzyme producing several smaller peptides. This viral protease is the target of anti-AIDS drug, viral protease. So this is what we have tried to, and this will be targeted by the drug. So which features essential for the success of these drugs? We have to see that it does not affect any other human enzyme. So a specific structure that inhibits only viral proteases, not any other protease which might be working in our body and will disable that and will result in malfunctioning of the body cells. We will end this video and we'll continue this in the next video, which will complete these questions.